Okay, welcome everyone to this session. We're really excited to start with this activity, the famous Polar Week that we usually host twice a year, and this is the second version. And my name is Renato Borrás. I'm the national representative of APECS-Chile, which is part of APEC International. And this is part of multiple activities that we're going to be holding during the whole week that are part of the Polar Week celebration. So you'll see that there is, if you Google it, you will see that there is some photo contest, and we have a few talks as well. And our activity, the one that we're in charge of, is called Breaking the Ice with APEC-Chile, and it's a series of talks that we are going to be doing during the week. And I'm glad to start this week with these two amazing scientists. So first of all, there is some ground rules of this seminar. We will be recording the session, but you're not allowed to record it. If you have any question, this is it's not just a talk. We're hoping that we are going to talk with each other. So you guys are more than welcome to interrupt or make us questions whenever you decide, especially along the presentation, or we will decide whether those questions can be addressed at the very end of the talk. But if you have any specific question about what the authors are going to be showing, you can lift your hand pressing the green button of the hand, and we will give you volume to your microphone. And also, if you don't want to do that, you can also write down the questions in the Q&A section, which is also down there. You'll see the option. This is a team, APEC-Chile. It's really, it's conducted by more than one person. So we're a team, and I want to thank that team because it's been a lot of work. Claudia, Estrella, Sambra, and myself. There's also some designers that have been helping us on the flyers as well as the website, and we really appreciate their help. There's also some other institutions that have been collaborating with us, especially when it comes to spread out the word, and the use of some platforms. So I really want to thank the Center of Climate and Resilience Research, the GAP, the Genomics Antarctic Biodiversity, and the Chilean Antarctic Institute, also Red Llama, which is part of the CR2. So thank you for them for helping us and supporting the work that we're trying to do here, trying to reach the young scientist, polar scientist. I wanna address also that the next talk we'll have before you guys leave, it's Earth, Wind, and Roots, Antarctic Teleconnections and Effect on Terrestrial Biota. Our guest is going to be Dr. Eduardo Castro, also Chilean scientist, and we're going to be having this conversation as well on Wednesday, the 23rd. Just remember for this that you guys need to sign in. So look out for the link and register in our social media, and also you can write us or go directly to our website. And today we have, in our first session of Breaking the Ice with APEC-Chile, we have Andrea, Dr. Andrea Piñones, and Dr. Luis Hückstädt here with me. I'm really excited for our two brilliant guests and really honored to have them here. They're both Chileans and have lately collaborated together from their areas of expertise, particularly in one beautiful paper that came out at the beginning of the year in Nature Climate Change, that really is a proof of a well-done collaboration and how this can work for a better understanding of Antarctica. So first of all, Andrea, she is a researcher of the Institute of Marine and Limnological Science of the Universidad Austral of Chile. Andrea is an oceanographer. She studied her bachelor degree in the Catholic University of Valparaiso, and then went to the US, where she obtained both her master and PhD degrees at the Old Dominion University in Virginia. Her PhD was already in Antarctic ecosystem, in which she studied the circulation of the Western Antarctic Peninsula and the implications for biological production. And in this path, she also became a very important figure in different Antarctic groups. She is a member of the Marine Ecosystem Assessment for the Southern Ocean, the MIASO group. She's an active research on the Dynamic Research Center of High Latitude Marine Ecosystems, the IDL project, and member of the American Geophysical Union, the AGU, of the Center of Oceanographic Research in the West-South Pacific, COPAS-SurAustral. And finally, but very important, she's been a member of APECS for quite a few years. Maybe you can tell me how many years already, Andrea, but I was lucky to actually meet Andrea in one session that was held in a conference a few years ago, where she was giving a very inspirational talk for young scientists, and I'm just thrilled and very thankful to be able to count on her again in today's talk. And also we have Dr. Luis Hückstädt. We met with Luis in California, in University of California, Santa Cruz, where I was looking for some help, desperate help for my PhD. And we ran into each other, and he's been helping me a lot with that. And it is actually where he still works as an assistant researcher at the Institute of Marine Science. Luis got his PhD and a couple of postdocs already with UCSC. But today also he's a young professor of the Department of Biology and Marine Biology of North Carolina, Wilmington, where he's actually living right now. Luis is a marine mammal expert, and I can tell that for all of us that work in this field, we have run at some point on Luis' papers or the couple of chapters that he write down, and essentially he quickly became an important player on the field. Luis has worked and collaborated in research involving all pinnipeds of Antarctica, except for ross seal. She has done some work on ross seal, but he's still in depth of working with that guy, which is very, very hard. And together with Andrea, they both published this nice paper that I was mentioning before. And he also was a part of a very important paper that came out in Nature also at the beginning of the year, which is called Tracking Predators Communities to Protect the Southern Ocean. So I think it's been a pretty good year for Luis, science-wise. And I'm really happy also to have him here. So we have two different approaches and ways of looking at the Antarctic complex ecosystem. With Andrea, we can revise how bottom-up regulatory mechanisms can shape the distribution of krill, which is a key species that shape the dynamics of the entire ecosystem. And with Luis, we can see how top-down regulation and the role and a little bit of the fate of the predators respond to those changes in the ecosystem as well. These two views combined result in a groundbreaking research, such as the one that I was mentioning before. And we, as APECS-Chile, are really proud to have them there. So I will give access to Luis. Luis for the screen, but Andrea talking, and we're gonna hear. Remember, you guys are more than welcome to continue making questions on our Q&A option, or you can lift your hands as well.
All right. So I hope you all, can you hear me well? Thanks, Renato, for that nice introduction. And I want to thank everyone attending this talk. Luis and I are very happy to be contributing somehow to this set of talks that APECS-Chile and APECS International have organized. So thank you guys for inviting us. And so, so if you can go to the next slide, Luis, please. So what I'm going to be sharing with you first in this first part of the talk is to tell you, like Renato said, tell you a little bit of how I'm looking at the Antarctic ecosystem. And as a physical oceanographer, I'm inclined to, of course, look at the ocean circulation, the ocean as a driver mechanism, as a driver of environmental changes in the ecosystem. So next, please. And so I wanted to start by showing you this classic conceptual diagram of the Antarctic Food Web, where we have Antarctic krill sitting right in the middle of the ecosystem, occupying this middle part and basically transferring energy from lower trophic levels to higher trophic levels in a very very effective way of transferring energy, right? And so, but also we have to think about that this Antarctic ecosystem is in a place where we have extreme weather patterns and ocean circulation is changing. And so we have all this set of environmental drivers that are affecting this ecosystem and that are affecting particularly krill distribution, abundance, and so this is what I wanted to show you today. Is next how the ocean control this lower trophic food web and how that relates to Antarctic krill. Okay, next. And so let's look at the Southern Ocean. So the Southern Ocean hosts this the most important current system of the world. The Antarctic circumpolar current is the one that connects the main basins around Antarctica, the Pacific, the Atlantic, the Indian Ocean, and in a way it separates, right, what happens in mid latitudes with the higher latitudes, but also is a mechanism of connecting all the basins, right? So we have the Antarctic circumpolar current going in an eastward direction around the Antarctic continent. And this Antarctic circumpolar current also transports a very important water mass, a water mass that is relatively warm and is nutrient high, is salty, and it provides heat and nutrients, and it's important in the heat and nutrient budgets that occur on the shelf around the Antarctic continent. And so next please. And so there are regions that are greatly affected or influenced by this Antarctic circumpolar current. And one of these regions, and one of the regions that I've been studying, is the Antarctic Peninsula, and especially particularly the western side of the Antarctic Peninsula. And so along this side of the peninsula, we have the southern boundary of the Antarctic circumpolar current running in a way parallel to the shelf break. And so the interaction of the ACC boundary with the shelf break makes this very interesting mechanism, ocean circulation mechanisms called the intrusions of circumpolar deep water, that, as you can see on the animation that is on the right side of your screen, how these intrusions of circumpolar deep water can connect the open ocean with the regions along the inner shelf of the West Antarctic Peninsula. And so we have intrusions of circumpolar deep water running along these deep bathymetric depressions and troughs along the shelf. And this circulation mechanism is basically bringing heat and bringing nutrients from the open ocean into the shelf. Next, please. And one of a large observation program, the Southern Ocean GLOBEC program. This was a multidisciplinary research program conducted in the early 2000s. observe that there are regions along the shelf in the West Antarctic Peninsula where you can find a high predator abundance. And these regions that, as you can see on the plot here, are very specific, and you can see a high predator abundance in specific regions like Crystal Sand, Lubo Fjord, and Alexander Island. And these regions are have a strong relationship with the circulation patterns that we observe along the shelf. And so using ocean simulation on regional ocean models, we were able to observe the relationship between these regions of high predator abundance with the circulation. And this is what I wanted to show you in the next slide. And so here we have a simulation of how the circumpolar deep water intrusions occur along the shelf, and how it connects the ACC with the inner shelf. And so what we observe here are the three regions described as a biological hotspot. And what we see is that the origination regions, so basically the particles, or you can think about particles, nutrients, heat that is coming into these specific areas are actually originating in regions around the area that we were studying. And what we also were able to find or to observe is that these, the main circulation pathways are the ones responsible in bringing these heat, particles, larvae, nutrients into this biological hotspot. So next. Also, using the regional ocean models and Lagrangian particle simulations as a toolbox for understanding the influence of ocean dynamics, we were also able to to observe or to study the connectivity of Antarctic krill populations along the shelf. And we we were able to observe that there are these two connectivity mechanisms that are important for maintaining and enhancing krill populations along the shelf. One is the local circulation pathways that can occur, like the ones that you see here that are marked as a local, and also the remote control of the circulation that basically is also important what is coming from upstream sources, right, into this study area. And so we went even a little bit farther and tried to understand how not only the circulation is important for krill in this area, but also try to understand how the environment is controlling early life cycles of krill. And for that, and I wanted to show you that in the next slide. We also did some individual-based model simulations to look at, or try to understand how, like I said, how the environment shapes the early life history of krill, and basically to look at how development, if development is possible along the inner shelf according to their environment, right? If it's a right habitat for the reproduction of krill. And we look at, and we did some simulations looking at the early desenescence cycle of krill. And yes, and we were able to observe that that along the shelf there are regions are very important for krill reproduction, and these regions are also consistent with these biological hotspots. So circulation is important for krill, and circulation and the local retention mechanisms are important for the development or for providing a right habitat for krill to continue development. So can you go to the next one? All right, and so, well, so these biological hotspots are located farther south in the peninsula, but there are other regions that also are suitable habitat for not only krill, but for top predators. And one of the nicest results from the US Antarctic Marine Living Resource Program is, well, they have several years of observations. So with those observations, we have been able to observe, this we can see you, okay, sorry. So we have been able to also observe this hotspot of high predator hotspot in the Northern Peninsula. And we also been looking at circulation as a driver for bringing nutrients and bringing heat into this hotspot in the Northern Peninsula. And the last slide I wanna share with you is this relationship, and now going more into the direct or a direct relationship between the lower trophic or the bottom-up mechanism as a circulation as a driver, and the top predators in a different ecosystem. This is a simulation of intrusion of circumpolar deep water, but now not on the West Antarctic Peninsula, but on the other side in the Ross Sea. And the red dots here are the locations of Weddell seals that were tagged with CTD sensors in the Ross Sea during 3 consecutive years. And there is, as you can see, the location of these red dots are very coherent and consistent with these yellow areas, right? In these yellow areas, red yellow areas are places where you observe the intrusions of circumpolar deep water. And so there is also a direct link between ocean circulation and higher trophic levels. And so with that, I wanted to pass the voice to Luis. Thank you.
All right, thank you, everybody. Thank you to APECS and APECS-Chile for inviting us to give this seminar. It's actually really cool to be part of this. So I'm going to switch gears a little bit, and we're going to be talking from the other point of view. So Andrea described her research, which has more of a top, bottom-up kind of a view. I'm going to talk about my vision, which is more of a top-down. So in this part of the talk, I'm not going to give you examples, two specific examples of my research. Instead, I'm going to explain a little bit about the philosophy. What is it that I study? What I study? So I thought that I would start by, if you ever work with marine mammals or seabirds or any marine top predators, usually explain our reason to study this species by telling people that these are sentinel species, and we can use these species to inform us about the status of the ecosystems, and we can actually use them to define baseline, so ecological baseline. So the first thing that we need to figure it out is what is it that we're talking about when we're talking about ecological baseline. It's basically information about the components of the ecosystems, right? So the ecosystems as a whole, not just this particular species, but I also want to know what their status is, their conditions, their connections, etc. The thing is that in a world that is changing so much, ideally we want to have all this data before things have happened, right? And this is because the only way for us to detect changes is to actually have something, a point to compare things against, right? So one of the best examples of this perhaps is the killing curve of CO2 concentration in the atmosphere, which even though it didn't start before the Industrial Revolution, we have a pretty convincing record that the CO2 concentration in the atmosphere has been increasing constantly since at least in the late 1950s up to today, right? So ideally, if we want to actually find out about how things are changing and what the status of the ecosystems is, we will have long time series that we can actually look at. However, the problem with that is that despite the fact that these long-term studies are actually very informative, it is really getting harder and harder to find funding to do this. So in this plot, what I'm showing you is the amount of money that has been invested in ecological research across the last decade, basically, for short-term studies or long-term studies. So the points in red, the line in red, which is showing a decrease, is how much the US government in particular has been investing in these long-term studies that are necessary for us to actually detect changes in the ecosystems. And you can see that it's decreasing as opposed to these short-term studies that actually are what the funding agencies are going for these days. And the other problem that we have, if we want to actually find out how things are changing, is that natural systems have been disturbed by us for millennia and centuries. So it's really hard for us to actually find out how things are changing. However, one of the good things about marine mammals is that they could potentially fill in this gap, and particularly in Antarctic ecosystems, because despite the fact that Antarctic ecosystems haven't been impacted as badly as other systems, they are changing. We know that the Western Antarctic Peninsula is one of the fastest warming areas in the world, but also in Antarctic ecosystems, the biomass, the abundance of these predators is particularly high. The diversity of fossils, for example, of true seals, is highest at the poles. So these species could potentially help us understanding what's happening with the ecosystems. And the reason for that is that these, in general, these marine predators, marine mammals in particular, actually integrate information through the entire food web. So this is because they have long lives. So something like a crab-eater seal, for example, probably lives to their 20s. A Weddell seal is probably getting up to the upper 20s or maybe even 30 years old. They cover large spatial scales. Think, for example, about whales, for example, how they can actually connect potentially the tropics with Antarctic ecosystems and back. So they actually are moving nutrients, for example, from polar latitudes all the way up to low latitudes where they actually go and breed. They're big animals, they have large bodies, right? So there's a lot of potential for something like carbon sequestration. They're endotherms, which basically means that they eat a lot. They actually need a lot of energy to generate to maintain their own temperature. So they actually are eating a lot of food. They're abundant, and this is particularly the case for Antarctic ecosystems. We have large abundances close to millions for several species, several millions for something like the crab-eater seal. And we have different life histories for these animals. We have some species that are residents. Think, for example, about all the APECS seals in Antarctica that they stay there through the winter, and other species that actually move back and forth. I talk about the whales, but also things like elephant seals, for example, so Ross seals that don't necessarily stay in the APECS system, but they actually move towards open ocean waters. And because most of them actually find their position on the trophic web is actually in the higher trophic webs, they actually have the opportunity to integrate information. So they are particularly good sentinels, right? They should be useful as species to inform us about the status of the ecosystems. However, if you actually look at the studies, this is something that is very well known for us, for our community, but across the oceanography world or the marine ecology, top predators are generally ignored. And this is probably because if you start thinking back about how oceanography started in the late 1800s and early 1900s, things started off by, first of all, the Challenger expedition in 1874, where they actually went out there and collected samples. And then you have things that started the kick-started the science, right? The opening of marine stations in Europe and the United States, for example, and then the zooplankton research is sort of what it started. But if you think about that, the common thread through all these events and through most of the development of oceanography worldwide was the fact that things were focusing on lower trophic webs. Lower trophic levels. So phytoplankton, zooplankton, that's it. So this, I was mentioning that this idea that we can use marine mammals as sentinels of the ecosystem is something very well known for us, but can we actually do it? So how much, in order for us to actually sell this idea and use the marine mammals to tell us something about ecosystems, we need to find out how much we know about the ecology of these species. And surprisingly, it's not very much. In this plot, what I'm showing you here is the number of papers that have been published on the issue of marine mammals and ecology during the last century, the 20th century, and whatever we have for this century. And you can see that it wasn't until the last 3 decades where we actually started working on this topic, when we actually start caring about the ecology of marine mammals and other marine predators. And this is particularly relevant because if we think about the historical context, what was happening during the 20th century was that we have depleted most of the stocks of several populations, several species of marine mammals. So this is just one example, but I can go through many of them, then it will be the exact same situation. In this case, these are Southern Pacific blue whales in blue, and you can see the sharp decline that these animals were having during the first half of the 20th century. And it wasn't until the late 20th century when we started to find out what was happening with them. So the whole thing is that the short answer basically is we don't know what their role is in the ecosystem because we haven't done the research. And then if you put it into context of the development of oceanography, you see that most of the knowledge that we've gathered about how the ecosystems work, how the marine ecosystems work, how the oceans work, how the ecology of the oceans work, came from a time where we have virtually no marine mammals. So it's not surprising then that most of these species were largely ignored by different efforts that put together a view of how we actually see the ocean. So in this case, for example, we have from the opening of marine stations in the United States, like Scripps and Goodsell, all across the mid-50s and 60s and 70s, where we have this sort of golden era of the development of oceanography, where we have all these multi-international efforts that put together a picture of how things work. Marine mammals were at the lowest. And it's not surprising that if we actually go to oceanography and how we actually understand the oceans, things are focused on lower trophic levels. So the NPC, nutrients, phytoplankton, and zooplankton model is perhaps the most useful tool for oceanography to describe how things work in the ocean and the system. But if you actually pay attention, things sort of stop at the zooplankton, so a trophic level of 2, and that's it. As these models have getting a little bit more elegant, perhaps a little bit more refined, they're starting to add boxes to these models to sort of understand how things work. One of them is the Tridus, for example. Fancy models actually have gone a little bit up on the trophic web and have included things like foraging fishes, like sardine and anchovy and so on. But that's basically where they stop. And new iterations of models actually put together ecological models like the NPC models with circulation models to understand how everything comes together in that part. But everything, as I was saying, is largely biased toward lower trophic levels, and marine mammals in particular have been largely ignored, despite the potential effort, the potential that they have to control entire ecosystem dynamics. And this is particularly relevant for Antarctic and polar ecosystems. So our view of the ecosystems, fortunately, has changed in the last couple of decades. This is taken from the book that I used when I was studying marine biology as an undergrad, as a master's student in Concepcion in Chile. And in general, this was how we thought about trophic webs. We have things focused on smaller, lower trophic levels. The only places where we actually consider the influence of marine mammals was actually in the Antarctic trophic web. That's the bottom panel that you have in there, where you can see it goes from diatoms to krill to whales. But that was it. Most of the other places, you wouldn't go into endotherms, for example, in the ocean. As things progressed during the late 90s and the early 2000s, the oceanography actually shifted towards even the smaller things. So that's when we look into the microbial food web and how that actually interacts with the entire food web of the planet. But the good thing is that now we're starting to understand that higher trophic levels are making a comeback. Marine mammals are making a comeback all over the world. We're understanding that they play a crucial role in the ecosystems of particularly high latitudes, because that's where most of these species weren't harvested to their lowest levels as everywhere else in the world. So they remain very important in these systems. And for those systems where marine mammals were actually mostly extirpated, there are several species are making a comeback. So we want to actually look into a holistic view of how polar ecosystems actually work. We need to understand that there's gaps in the knowledge and we need to catch up to that. So we need to understand how different species interact with each other and how they interact with the system and how they actually work internally. So that's what my research is focused on. I work mostly on this sort of interaction between physiology and ecology, and these are the different aspects that I'm interested in. So you need to understand how the individuals are actually operating and what are their demands, energetic demands, and how much they consume. But also I'm looking into the ecology of these animals, how they actually fit within the trophic webs, how they're moving, and how they're actually using the environment. And if you see there on the right at the bottom, animals as environmental sensors, that's sort of part of the research that kick-started this collaboration with Andrea, because we are looking at the animals not only as species that can tell us about the status of the ecosystems, but actually we can use marine mammals as sensors of the ocean. We can put sensors on these animals that actually are going to be measuring things like temperature, salinity, oxygen, fluorometry, and inform the oceanographers about what's happening with the ecosystems at these lower trophic levels.
Yeah, Luis, I want to like hop in this, because you mentioned the collaboration you have with Andrea, and maybe you can tell us, I mean, what Luis is talking, for those of you who don't know. Luis and Andrea published a paper in Nature Climate Change called Projected Shift of the Foraging Habitat of Crab-eater seals along the Antarctic Peninsula. And well, Luis, could you tell us a little bit more about it, because it might be helpful for the rest that haven't been.
Yeah, that's perfect. I actually have the slides following up. We're actually about that. So,
oh, nice.
Crab-eater seals are perhaps one of the best examples of how we can use a species of higher trophic level to inform us about what's happening with the rest of the ecosystem. And this is due to the fact that they are highly specialized on krill. Pretty much that's the only thing that they eat. They can eat fish, but about 95% of their diet is Antarctic krill. So if we think about that, where they go is where krill are. So as part of the Southern Ocean Globic project, and then a different project, as Andrea was mentioning just now, we put out tags on crab-eater seals along the peninsula and look at their movement. And once we started looking into that, we decided that it might be interesting to see how they actually are using the environment, what environmental cues they're using. So we built together habitat model for the species. But it turns out that most of the environmental data that we can actually use as covariate for this model come from sources that are not particularly well developed for Antarctica. So satellite imagery, for example, and the knowledge of what's happening in the water column is not as developed as we would like for things like a habitat model for a species. So that's when the collaboration with Andrea started, because they had a model at Old Dominion University when Andrea was doing her PhD. They have a three-dimensional regional ocean model system put together for the peninsula, so we could actually extract data from that model to fill in the gaps of the covariates that we needed for our habitat for the crab-eater seals. But not only that, we could also run this model under different conditions to see what was going to happen in the future. So we were able to actually look into projections of what the habitat of the crab-eater seal will be for the future. So this is a little animation where you can see on the left, you're going to see in the predictions for current habitat, and on the right, what's projected for the end of this century. The yellow colors actually show high importance of the habitat, and the blue colors basically areas that the crab-eater seals are not going to use. So what you see here, and this is a monthly comparison, is that things are going to be shifting in the future. Crab-eater seals habitat, basically, which should be defined for like where krill are, is going to extend offshore and it's going to shift south. So what we think is going to happen is that as things actually progress and the sea ice season is shrinking and the temperatures increase and the intrusions of sea comparative water get intensified along the peninsula, the bulk of the biomass of the krill is going to shift south and offshore. So it's going to be harder for these species to reach the krill. So they're going to go from a situation like this where you effectively have the overlap between crab-eater seals and the prey. Crab-eater seals need sea ice to hold out. They're going to hold out exactly where krill are. So literally all they have to do is just roll off the float, dive, get krill, and come back and hold out. And we think that's the reason why they've been so successful. To a different system, to a different situation where krill is probably going to be expanded offshore. So they're going to have to now travel, invest energy into traveling to get to where the seal are. And on top of that, the plot on the that is shown with the letter C is that there's also a different research that are showing that the density of krill is also going to decrease. So it's not only the fact that they're going to be advected offshore, but also they're going to decrease. So that basically effectively poses a challenge because there's going to be a divergence between the ice where that seals need to hold out and where the krill are.
Great. I have a ton of questions, but we're going to go with, we have a couple of questions here. Before I forget, because it's kind of tied with what you're saying, Luis. Andrea, you made very clear, I had a question about the CDW instructions, but you made it pretty clear that how important are. But sometimes you get confused with people that is not oceanographers, because you were saying that they provide nutrients and also heat. And you would think, for those that we don't work on that, that heat is the opposite of good. Especially when you start thinking like, hey, climate change, et cetera. Could you specify exactly why would those will kind of shape the heat budget on these areas and therefore the nutrients that you came up?
Yeah, so when you look at krill, for example, and you look at the early life cycle of krill, the early life part of history of krill, actually these warmer places are regions that are somehow provide a good environment for the ascent cycle to complete in less time and in a shorter depth, basically. And so these intrusions getting into from the open ocean into the particular regions along the shelf would enhance or be positive for the descent-ascent cycle to complete in regions. So you don't need very deep regions for this descent-ascent cycle to be successful. But that's only one part of the good part of the heat, right? Because also you have to take into account that there are other environmental variables that are very important for the early life cycle of krill, which is basically sea ice formation. And we know from observations and from trends that, and from projections, that the sea ice timing has been changed, has decreased in the past years, past decades. And that is not good for the early life development of krill. And so that's basically the good and the bad part.
Yeah, I actually have another question. I have one for you, Luis, but this one is very related to what we just talked with Andrea, so I'll go with that one first. Mike is asking, hey Andrea, this might be a basic oceanography question, but why is the circumpolar deep water protruding into the colder and closer to Antarctica waters, and not the surface water above the circumpolar deep water, assuming that the surface water is warming? You can, I think you can, yeah, you got it.
All right. Yeah, I think I understand the question, but maybe I'm not. And if I don't answer correctly and you don't understand me, please, you can explain me better the question. But so the circumpolar deep water is a water mass that is located below 200 meters. The core of the circumpolar deep water is more or less around 300 meters in the open ocean. And so these intrusions occur along deep bathymetric depressions and canyons. And they find their way from the open ocean into the shell through these channels and through these depressions. So that's how you get actually warmer circumpolar intrusions into the shell. And then local processes on the shell make the rest. They mix strong winds and mixing and bring these warmer waters and nutrient-rich waters into the euphotic layer, so the primary producer can use it, right? So it's a combination of these intrusions with local physical drivers along the coast or along the inner shell that do the rest for the nutrients to be being brought up to the surface.
Okay. Mike, if you have any other questions, you can also raise your hand. Yeah. Maybe it's easier than texting in case you have another question regarding that. Luis, this is, for those that we work on biologging, this is a typical question that we usually get from audience. And I think it's very important that you clarify, because there's people, I mean, there's still a little bit of afraid about this. What is the impact on the behavior, and specifically on the reproductive interaction of the organisms that are used to carry those CTD sensors? So it's basically about the instruments itself and the effect that the instruments can have on animals.
Yeah, it's actually a really good question. As Renato was saying, we get asked that question all the time. The short answer is either 0 to negligible. And the reason why we say that is because there's several factors you have to consider here. The first one is the impact that the tag itself could actually have on the animals. And the largest instrument that we're ever going to put in an animal is usually about 450 grams. Whereas these animals are at least 300 kilos up to, the largest elephant seal that I've ever put a tag on was 800 kilos. You can actually see that the impact in terms of the mass is less than 0.1% of the mass, right? There's been a lot of development on the technology to actually make these things as hydrodynamic as possible to reduce the source of potential turbulence that it will have on animals. But as a matter of fact, we put the tag on the head for several reasons, but one of them is that it's not actually going to affect the hydrodynamics. If you think about a seal moving through a field of water, the widest part of the seal actually is the shoulders, and that's where you're going to have turbulence flow of the major resistance, right? The tag actually is so small that it's actually not increasing the drag that these animals are going to facing as they move through the water. So in terms of the impact of the tag, the technology has reached a point where we've been able to miniaturize it to a level where the impact is 0, basically. And there are efforts to make it even smaller. And that's one of the reasons why this fast development of the technology has been very beneficial, not only for us, but also for the animals. We still have some limitations in terms mostly of battery power. That's one of the things why the tag still has to be a little bit too big, but as the battery technology is actually increasing worldwide, we're actually going to be able to move towards the different sources of energy or different sources of energy storage. And the other thing that we have to consider is that when we do this, of course, our first priority is to keep the animals safe. We don't want to cause any repercussions to the animals. We don't want to cause any consequences to the animal, because also that would affect the kind of conclusions that we can draw from the data that we're collecting, right? So if we affect the behavior of the animal, we couldn't be using those data to inform us about either the environment or the ecology or the behavior or the physiology of the animal. So we make sure that the impact are reduced, and we also make sure that animals that we're going to be using for that, we're so careful. We usually go for animals that are looking really, really good. So if an animal is looking at least a little bit compromised for whatever reason, say for an animal, a crab-eater seal that could have been recently attacked by a leopard seal and it has a scar, we will never touch that animal, right? So we're making sure that the animals that we put tags on actually are at their best health and best body condition before we even try to get a tag on them. And in terms of reproductive output, we haven't directly addressed that in a species like the crab-eater seal just because they're so difficult to find out, but there's several papers that have done something like that on a bunch of different species, smaller than crab-eater seals, larger than crab-eater seals, and there's no effect. If anything, probably because we're actually going for the healthier animals, animals that actually have had a tag usually do better than animals that haven't had a tag. So say, for example, we put a tag on an elephant seal that goes out to sea for 8 months and comes back. We know, because we measure enough animals by now, that animals that we've actually handled, we put instruments on, have gained more weight than animals that haven't, than the average of the rest of the population.
That's interesting. I have a classic question as for both of you, but it's probably the most important that we can also draw from your results of the paper that you guys collaborate. What can we expect for the future? I understand the scenario for the crab-eaters specifically on this example of the paper, but this might have a couple of different questions if I address it to Andrea or Luis, depending on how we see the ecosystem. So in the case of Andrea, we're usually more familiar to the problems that the loss of ice would have on the impact that would have on krill, but now you're telling us something that is perhaps less more common in just a regular Antarctic literature that we non-oceanographers tend to read, and it seems to be a very important. Have you been seeing the influence of these deeper waters in the generation of these hotspots? Are those is there any projection of where are they going to go, or have you seen in historical records of how they have already changed in the past? How big is the time lapse between those changes, etc.? We know that ice loss is going really fast, but what to expect on this ones?
Oh, that is a very interesting question, and I also I asked myself that also, and so there are a few scenarios I think that we can look at, and like if we so these regions, these biological hotspot regions, are primarily there is a large abundance of predators in these regions, and they are there because they can feed, right? Because they have food resources to prey upon. And looking at krill as the main or as the key species of the Antarctic ecosystem, so in this hotspot, not only you find predators, but observations also show that are regions with a high concentration of krill, and these are observations done during multidisciplinary research programs, right, that have been like going on for years and years, and looking at the same places, and always finding these predators in the same regions. And so if we remove, I would assume that if we remove the krill, or there are not the right conditions or environmental conditions, or the environmental conditions along the peninsula change drastically, like loss of sea ice and circulation, perhaps circulation pathway shifts, we can assume that if we remove the krill, I would guess that the predators either look for other food resources, or I mean, change their diet in the same place or just move to some other place to feed, right? Where the krill goes. So yeah, so we have done some simulations looking at Lagrangian particles and looking at like how the retention will change in those regions. Like we haven't published this, but so there are the simulations have shown that there's going to be a shift of these biological hotspots a little bit south, and these. Okay. This is mainly because of the environmental conditions will be optimal farther south and farther north.
Yeah, but very consistent with what you guys found on the paper as well, like the projection of the shift on to south and ocean-wise. And then same, like Luis, you guys have shown us in the paper what could be the problems for one specific species, the crab eater, which is we that work in marine mammals know that it's basically all they eat. But we have another guys here that are also very dependent on krill, and we have, for example, penguins and fur seals around that also rely heavily on these krill patches, but they do have a very a little different, which is the fact that they are central place foragers, which mean that they have breeding sites from where they will go and perform foraging trips to later go back and feed their offsprings, basically. Do you have any speculation or perhaps some information of what could happen with these species too?
Yeah, and we actually what
is different than crab eaters?
We actually touched on that on the paper as well, because as you're saying, it's not only, we're trying to use the crab eater seal to tell us about what's happening with the ecosystem, not only with the crab eater seal, and that was the whole point of that. You see this, this is the figure that tells everything about how those central place foragers are going to be affected. So the South Shetland Islands, which I don't know you can see the mouth, are in this part of the tip of the peninsula. Our model is actually showing a decrease in the, so there's very few places where you can actually see decrease in the foraging habitat of crab eater seals, aka krill habitat, except for the area around the South Shetland Islands, where there's a large concentration of colonies of fur seals and penguins, right? So this area, this is particularly relevant because it happens during a time of the year when those animals are actually there breeding. So during the summer in Antarctica, that's when our model actually projects a reduction in the krill habitat along the South Shetland Islands. So the problem with these species is that they're bound to a colony, right? Crab eater seals, they don't have colonies, so they can follow the krill and they're probably going to shift south and they just stay where the ice is. These species, on the other hand, they have to come back to their colonies, and the impact that we're forecasting for these species is actually higher than for other things like whales, for example, or crab eater seals who can actually move. These species will potentially have to start colonizing new lands as things move southward, and they're probably going to have to follow that. But that process is a lot slower than just moving with the ice, as crab eater seals could do, or humpback whales or minke whales could do. So the impact for these species is particularly important, because we don't think they're going to be able to cope with this reduction in krill as much as easily as other species. So the only opportunities that they'll have will be A, switch to a different prey, switch to fish, for example, myctophids, which are probably more energy dense, so higher reward for them if they actually catch them, but they're also harder to catch and not as abundant as krill. So there are levels of plasticity for all these species that we don't really know what's going to happen. Penguins generally do really well, or they do prefer fish in some other areas of Antarctica, in the Ross Sea, for example. But krill is so abundant, there's so much of it, and it's so easy to catch that, of course, a reduction or a shift in the distribution of krill is going to impact all of these species.
Yeah. Yeah. and it's making a comment about that. So crab eaters will turn into central place foragers. Like if you start tidying out, you're eventually.
Effectively, that's what we think is going to happen. It's not a central place for you per se, because they're not going to be going back to the same spot. But the ice is probably going to be landlocked, right? So whatever ice they're going to need to actually hold out, it's going to be closer to the coast, where krill is going to be farther away. So instead of, as I was saying earlier, just roll off the ice, dive, get krill and come back, they're going to have to now travel horizontally to where the krill are, dive, and then come back, because they need to rest. And also, it's not only the problem that they need to rest, they also use the pack ice as a refuge, because they're falling prey to killer whales and leopard seals. So the more time they spend in open waters, they can actually fall prey to killer whales. So it's a not so good situation.
Andrea, we have a couple of questions for you. And this kind of link with another one that I had. What happened with adult krill in relation to the change that the Antarctic ecosystem is undergoing in recent decades? You have observed any change of size of adult krills?
Yeah,
you were mentioning that most of it. You were mentioning that most of it's occurring in early stages of krill. What happened? Where is the adult krill? This picture.
Well, I would love to answer that question, but actually, I don't know. So I'm not a person that works with krill. So I unfortunately I can't. I don't have the answer to that, but probably we would have to look at the latest studies done in the Amalar region or Area 48.
But when you guys were sampling basically the shelf break and probably the platform, the oceanic platform, you were finding basically most of it early stages?
All right, so during the observations that I shared with you and the results from the Southern Ocean Globe program, so the observations were done during the winter time, because we were very interested in finding out what was happening with the winter conditions and how the krill can survive and can use different mechanisms to survive during winter. And so there are observations and results obtained from the distribution of krill, not only Antarctic krill, other krills along the shelf. So basically the adult krill is pretty much found during the summertime. Is found and gravid females are found along the shelf break in like off-shelf regions where they can lay their eggs. They do this seasonal migration, krill, and so during the winter time and the observations that we obtained during the Southern Ocean Globe program, we were able to find different life stages of krill, larvae, Fusiliia stages along the shelf and along parts like along the inner shelf, and different parts around Margaret Bay in the deepest place. Yeah, I don't know if was that your your question, right?
Yeah.
Okay.
Yeah, it does, this one is also very interesting. It's also for you, Andrea. What is the impact on the system of freshwater glacier originated related to the CDW in relation to the heat and nutrient sources? Like the input of freshwater originated from glaciers that is also kind of messing up with this, probably messing up, I don't know, with this CDW intrusions as well. Is it changing the temperature? Do you guys know if there's any pattern in places where you have more intrusions of fresh water than others?
So the question is, like, what's the balance, like the balance between the heat that is transporting the circumpolar deep water into the shelf ecosystems or the shelf system, or the inner shelf, compare with what is coming out from the glaciers? Like, I'm sure there are some studies.
Yeah.
So I would say that both are a source of nutrient. I think the question is related to nutrient source, right? I think both
the
are
nutrient sources.
Mm-hmm. So both are a nutrient source. So what is coming out, or what is the fresh input source that is coming out from the glaciers, or the retreating glaciers, or the freshening, what is called the freshening, or the impact of this fresh water along the shelf systems, the embayments and the fjords in this case along the peninsula. I think these are also input sources for nutrient, micronutrient like iron, basically. And then we have the impact of intrusions of circumpolar deep water transported heat and nutrient, mostly macronutrients into the shelf regions. So I think there is an influence of both in providing nutrients to the shelf environment. What is more important than other? I think both are important.
Who knows?
CDW can also transport because it interacts with some places where you find bathymetric depressions, can also somehow transport iron, and it's been shown in other regions, perhaps not the peninsula, but in the Ross Sea, that it is also a source of iron for the shelf ecosystem, at least for the Ross Sea. So I think you have both being an important source.
Yeah. Okay. We have another question from Caroline. It's for Luis. And it's kind of following up what you were explaining about the other species, that for those that make colonies, do you think that it will start like breaking up into tiny colonies and become more flexible somehow? We have some, it's probably associated to how specialist can be those individuals, and make and kind of related to some areas of, because it's getting tied down more and more, you might have small colonies with different strategies, which is.
Yeah, and then as resources get limited, right, you'll have higher competition, higher interspecies competition, right, for the same resources. That's a good question. I don't think we can actually predict what's going to happen. I think as a general trend, what you'll see is a shift of the colonies, and as terrain gets exposed, so land gets exposed as the ice is melting, chances are these species are gonna start colonizing farther south, right? The whole point, the problem will be that how fast can they actually do it? Can they actually do it fast enough to keep their population levels or not? None of these species are in danger, right? So if we're thinking about gentus and chin straps, even Adelies, which in the peninsula are sort of decreasing, as a whole, the populations Antarctic-wide, they're actually pretty healthy. So I don't expect any of these to have a major effect, like the species is going to be going extinct. Eventually, that will happen if they're not capable to keep up with the changes, and that's the main concern that we have: how fast they're going to be able to react, how fast they're going to be able to colonize new areas that are getting exposed, where they can actually build their nest and have chicks or pups, for example, in the case of Antarctic fur-seals. But we already know, for example, that there's a new colony of Antarctic fur-seals that is pretty far south on the peninsula that we just discovered last year. So these species could potentially expand as the ice retreats and move towards higher latitudes. One of the best examples are the elephant seals, for example. Elephant seals, which we think of them as sort of ice associated, maybe they don't like ice so much. We actually seen elephant seals. I don't know if you guys can see my mouse, but we see elephant seals getting this far south, like to the very tip of Alexander Island, and actually foraging in that area. And some of them go deep into fjords that actually look more like crab eater seals. So it's not like species are just going to go extinct, everything's going to be negative. Hopefully these species have the plasticity, the wiggle room to actually adapt, adjust their behavior to how the conditions are. But there's going to be an impact no matter what, because things are moving so fast that even though they do have some plasticity, chances are they won't be able to adapt that fast.
Okay. Yeah, we're kind of running out of time, so I kind of want to address this question for both of you guys. I'm familiar that you got in touch in the GLOVIC, I'm going to call it initiative. Originally, you guys started on your expertise areas on about kind of around the same year in 2008, but and I did mention all the groups that Andrea is involved, MIASO, GIU, the Ideal Project, etc. And I didn't even wanted to name the numerous groups that Luis is involved as well, such as the Birth and Marine Mammal Expert Group of SCAR, etc. And a month ago, we have the SCAR online meeting, and I noticed that some of the great collaborative works came out of these groups of expertise. But I don't know, and I really, I think it's very useful for our audience to start knowing how can they get involved, or how did you guys start getting involved in those groups? And probably also to know a little bit how are the responsibilities of becoming part of these groups. I don't know, whoever wants to start, it's fine. Luis or Andrea.
Andrea goes first.
Okay. So how I got involved, I think through collaboration that started early on in my career when I was a PhD student. I think the key was when I was a PhD student, I participated in many of the international meetings, especially the SCAR meeting was one of the ones that I was the opening door for me to get into know a lot of scientists working in the same, at least in the same area of research that I was doing or that I was involved. A key aspect for me was to become part of APECS. This will sound funny, but yes, I think, and this is something that I encourage young researchers to do, and the people that work with me that are starting their career in Antarctic science, is that to subscribe and to get involved in APECS. This is a very good way to get to know people and take the opportunity to do all these online talks and posters and webinars and so on. Yeah, and now, well, my responsibilities are trying to, be attend meetings, well, now virtual meetings, and yeah, basically that.
How about you, Luis?
It's kind of similar. I was lucky enough that I got, when I first went to the States, I wasn't supposed to be doing Antarctic research. I was supposed to be working with sea lions in Chile, and that fell through. But my advisor had an NSF project for Antarctic research with no students participating in that. So I jumped into the opportunity. I started doing my dissertation based on those. And as you mentioned, Andrea and I, for example, met at one of these workshops that was part of the GLOVIC in 2007, 2008. So I started being part of all these meetings and being sent to different workshops. And yeah, as Andrea say, I've been going to the SCAR meetings since 2008, I think. And all that opportunity to interact with people has been great. I saw that the great Monica Mulberry sending a chat with the with the link to the EGBAM group. Being part of these projects is actually most of the programs and groups, expert groups, and different initiatives within the SCAR are very open. So you can actually be part of those, attend the different meetings that are going to have during the SCAR Open Sciences or the Biology Symposium, and then slowly get to know people and also participate. If you volunteer, for example, that you want to be part of one of the initiatives that they have, that of course will put you in touch with several folks from all over the world, and you can actually start working with them. So the scientific community involved in Antarctic research is perhaps one of the most open and collaborative communities that I've ever been part of, and it's relatively easy to take advantage of that and be part of all those efforts. And I'm not talking about being established researcher. As a student, you're more than welcome to be part of all those different initiatives and groups.
Okay, that's great. Are you, just a quick answer, because we're running out of time, but are you guys, I assume that the collaboration ended up well, otherwise we wouldn't be able to have them both together here. Are you planning on any future collaboration? Are you guys planning on any future Antarctic collaboration, and any plan on the projects that you're currently applying for?
Yeah, we certainly are thinking ahead and continue our collaboration. Just quickly, just tell you that with Luis, we're hoping that our proposal gets funded to keep continuing looking at now tag Weddell seals along the inner shelf in the Northern Peninsula. So hopefully we can continue our collaboration through that project.
Yeah, and for my part, I have two different projects that I'm also hoping to get funded. And for both of them, I'll be working with Andrea. I'm going to continue on putting these instruments that can measure salinity and temperature of the water column in Antarctica. And I know for a fact that those data, Andrea will be very eager to work with. So I have her as a collaborator for both of those initiatives. And eventually, once I don't have a permanent position, but once I get that, for example, we can actually do more research together. But yeah, we have plans to continue on working in Antarctica and also in Chile, actually.
Oh, that's great. Well, that's great. It's great to hear from both Chilean scientists as well. This is really great for us, APECS-Chile, too. And I really want to thank you guys. If you want to follow up these collaborations that the guys are talking, you can also follow them on their Twitters and on websites that they have. And we also, well, first of all, again, Luis and Andrea, thank you very much for your time today. It means a lot to us. It's a tough day for Chile, considering that we're coming out of our Independence Week and we're still a little bit shaken. But we're sure once we share this on our websites, we're definitely gonna hear back more from other scientists around the world. So thank you very much, and I will also like to invite you guys to the other activities that Polar Week is holding this week. And specifically remember, you guys, that we'll see each other again on Wednesday with Eduardo Castro. And you need to sign in, so don't forget to register, and you can find a link in our social media and website. So thank you very much, everybody, for attending. Everybody. I don't know if you guys want a final word.
Thanks, Renato, and thanks everyone there on APECS for organizing this. It was a pleasure.
Yeah, ditto.
Great. Just remember that APECS-Chile is part of APEC, the Association of Polar Early Career Scientists worldwide. And I'll see you guys on Wednesday again, and have a great rest of the week. Thanks again, and see you guys.